Hey folks, welcome again to Anatomy Physiology. I know you can't wait to get started. Neither can I. As soon as we start, we can finish it up. We've got two more chapters, chapter 22 and chapter 24. This is on the lymphatic system and immunity, okay? Got a lot of objectives, and I'll try to cover most of them here. I'm going to try to lecture in shorter segments. Maybe I can cut down on the number of videos that are posted, but I don't understand how some of it works anyway. Doesn't really matter. I'll post this first right here, then I'll post this when I send it out. Define antigen and a pathogen. First objective right here. Antigen and pathogen. I've got the word pathogen over here, but let's talk about an antigen first. An antigen is anything foreign to the body. Anything foreign to the body. For the hell of it, you can take, let's say that you were to make a cut right here, and I were to turn this little sliver of tissue upside down. The cells on the stratum corneum, you may remember that one, are different than the stratum basal. So anytime a cell changes, it is foreign to the body. Red blood cells change, they age, they're foreign to the body, and they are removed. So an antigen is anything that's foreign to the body. A pathogen is a disease-causing antigen, an antigen that causes disease. And God Almighty, we know about the Corona-19 virus. We don't know all about it. We know more about it than what the hell we did, okay? Now let's talk about the formation of lymph. I'm not going to draw all of it up here, not all of it, but I will go back to the capillary network. Go back to the capillary network just for a few seconds. Of 100 molecules that go into the capillary network, the blood flow is going to go in this direction right here. 95 of them are returned. 95 of them are returned. The 5% right here is lymph. So go back and take a look at your notes on the formation of lymph, okay? It's this extra interstitial fluid that is not returned to circulation. But 95% of it goes back into circulation, okay? Now the reason it goes back into circulation, we'll cover that in this last chapter here, is because of the presence of albumin. Albumin is a very large protein made in the liver right here, called the osmotic regulator. As this fluid goes out, the contents left in there become more concentrated, and there's an osmotic pressure that causes the drawing of the water, of the fluid, from the interstitial fluid, from in between the cells, back into circulation, okay? Now, if you have a liver disease, if you have hypoalbuminemia, you will get an increase in lymphatic fluid. You will get maybe an increase in interstitial fluid, you may get edema. We'll cover that in a few seconds. But anyway, this is the formation of lymph. The structures in the lymphatic system and the 5 lymphoid tissues or organs are going to be the same thing. So we're going to combine those two right here. My page is on 22-1. Lists for me some structures that are in this drawing right here. Lymph nodes, spleen, thymus. Okay, those are 3 right there. And pick out something else right there that we have right there, okay? Give me 5. Okay, what stimulates, regulates the movement of the lymph? Well, excuse me, I've already skipped one right here. 5 lymphoid tissues and organs. I'm going to touch on associated lymphoid tissues, okay? Now, ALT is what I'm after right here, ALT. Anytime that you get an aggregate, anytime that you get a bunch, an aggregate of B cells or T cells, you're going to get associated lymphoid tissues. You may not have a capsule around it, it may not be an organ by itself, but these are going to be associated lymphoid tissues, okay? Associated lymphoid tissues. We find them in the gut. Believe it or not, we call it GALT. We find them in the respiratory tract. RALT, R-A-L-T, respiratory associated lymphoid tissues. Gut associated lymphoid tissues, okay? Now, I want to include that as far as lymphoid tissues and organs, okay? Now, what stimulates or regulates the movement of lymph? It's the same thing as moving the fluid in the veins. It's the presence of one-way valves and muscle contraction. It's the presence of one-way valves and muscle contraction that stimulates the movements of fluid in the lymphatic system right now. Now, what happens to the lymphatic fluid? Something's got to happen to it. It's got to get back into circulation. And it is dumped back into the vena cava right next to where it goes into the heart. But there's two large structures that do this right here. And we're going to get a picture of, oops, too far, of this right here. And I'm on figure 22-4. 22-4 shows the two ducts that carry the lymphatic fluid back into circulation. One is the thoracic duct. It carries about 80% of the fluid back in. The other is the right lymphatic duct. So I want those two in your answers. So those two answers. So we have to return this fluid back into circulation. It cannot build up in the tissues. It cannot. So this is a slow moving system. It may take about 24 hours for this to occur. Lots sort of what the venous return is, okay? Because it's only 5%. Maybe that's the reason. Okay, but anyway, the three types of lymphocytes: B cells, T cells, and natural killer cells. B cells, T cells, and natural killer cells. All right? We'll take a look at the different functions of these right here. A B cell, a T cell, and a natural killer cell. A natural killer cell. Those are the types of lymphocytes that we have right here. List the 4 types of T cells and their function, okay? 4 types of T cells and their function. Now I don't want to miss any out. So I'm going to take on the 4 types of T cells that we have right here are memory cell. We have a cytotoxic, C-Y-T-O-T-O-X-I-C. Cytotoxic. For a long time I thought a cytotoxic and a natural killer cell were the same thing. And maybe they derive from each other, I don't know. But we separate them anyway right here. Cytotoxic over here, suppressor, and then I've got a helper cell. Huh? Now, as far as our function right here, anytime that we stimulate our immune system, we're going to build up some immunity, and it's mainly going to be the presence of the B cells, but that's not, our focus is not on the B cells now. Our focus is on the T cell right here. When the T cell is exposed to an antigen, it's going to divide and produce a memory cell. The memory cell is going to cut down on the response time. That's a function of it. How long does it last? 6 months, a year, 2 years, 4 years, somewhere in there. It's going to be different for different antigens that we have right here. So the memory cell cuts down on the response time. All right, I know more about the B cell and its response time, but I know that there's a memory T cell that we have here. So cut down on the response time. Cytotoxic is versus cancer cells. Cytotoxic is versus cancer cells. All right, that's what I want you to write down right now. Suppressor right here may surprise you that anytime we turn the system on, we got to be able to regulate it. We got to be able to turn it off. It's kind of like the blood clotting scheme. The plasminogen to plasmin to break the clot down soon after it starts forming, the fibrinogen to fibrin to make the blood clot, the last stage in the blood clotting scheme right there. And then lo and behold, we got to remove the clot. So we have plasminogen to plasmin. This is no different right here. So we have a system in mind, in place to suppress the function of the T cell anyway. Because we do not want this damn system activated all the time. If we're not exposed to antigens all the time, we don't need the system turned on all the time. Because once these boogers are turned on, they're going to attack something. We do not want them to attack our healthy cells. It does happen, okay? Then we have a helper T right here. A helper T really is associated with AIDS, the HT, the T sub H right there, the helper T right there, associated with AIDS, and that'd be fine right now. It's going to stimulate the production of antibodies. That's the function of the helper T. Stimulate the production of antibodies. Okay? Now, lymphopoiesis is a production of the red blood cell. It's erythropoiesis, but it's still got poiesis in it. Anyway, lymphopoiesis right here. So what I want out of this is, where are these cells produced? Where are these cells produced? You may remember way back when, not that long ago, last chapter right there, we talked about hematopoiesis and the production of all the blood cells. Well, this is one of the blood cells. And on the right-hand column is a lymphoid stimulating colony right here. So the very right-hand column, these are separated from the myeloid. Myeloid is all the other blood cells. Lymphoid would be the presence of the lymphocytes that we have right here. We have lymphopoiesis. We're going to start off with the bone marrow right here. The bone marrow. Now, these cells are very immature when they leave the bone marrow. They're going to go to the thymus, especially the T cells. They're going to go to the thymus and mature. Thymus. Selection and maturing. This is a function of the thymus gland. The thymus gland that we have right here, okay? I don't have a thymus gland anymore. It's been gone for years. Yours is about half of what it was 4 or 5 years ago, okay? It's the way that it is. So the thymus gland selects and matures T cells. That's its function. That's what I want you to write down. Thymus selects and matures. Now, if a T cell does not pass a test, if it attacks your own healthy body cells, it's going to be eliminated because you do not want that cell line produced. So selection and maturing are the function of, is the function of the thymus gland that we have right here. Okay. Now, let's go ahead and list the lymph node for maturing of B cells. But that's okay. I'm not going to separate maturing from B cells. Bone marrow, thymus, lymph node. All right, let's go ahead and list the lymph node. We'll talk about the area of the lymph node that has a concentration of these cells anyway. Okay, I'm going to turn it off for right now, and maybe this will be one segment, and that'll be fine right there. I'll be back in a few minutes.